So I'm going to talk about dual agent dose escalation studies in phase 1 trials. And we developed a model-free approach. We've worked a lot of model-based approaches as well. And so I'm going to describe a method that we call the product of independent beta prior probabilities dose escalation, or acronym PIPE. And we published this early last year, Mike Sweeting and myself. And we've tried to get this implemented. So we're trying to develop something a bit simpler that people might implement. There's a real problem in phase 1 trials that a lot of trials don't use model-based designs or don't use statistical models or statistics. They just use rule-based designs. So one step to move away is try and develop something that is simple to use. So we tried to get this, or it is being implemented in a trial at UCL. We've also got a collaboration with Roche Pharmaceuticals, and Chris Harbron has taken the model and developed an R-Shiny app, and they're implementing it in one of their trials. I can show you some of the simulation results so we can compare to other types of dual agent designs, and I'll describe some of the software because we've got various implementations of it. So in phase 1 trials, a common goal is to find the maximum tolerated dose, or in dual agents, it could be dose combinations, more than one. A lot of the methods out there just look at a single dose combination that they think is the maximum tolerated dose combination. With our method, we're looking at multiple at the single time. So the usual thing is we give doses to small cohorts of patients. We see how many dose-limiting toxicities you observe. Is it safe to increase the dose? You make that sort of decision. The definition of the MTD is quite a precise thing. So it's about, or in some cases it's precise, it's about the probability of a DLT at a given dose. This dose might not be a dose you've got in your treatment selection. It might be a theoretical dose, but it usually equals some sort of target toxicity level. And these are generally selected as 1 in 3, 1 in 4, 1 in 5. They're kind of dependent on which disease you're looking at. And the 3+3 design is the rule-based design that a lot of people use. And that doesn't actually define any of this sort of terminology. It doesn't have a target toxicity level, and it doesn't define the MTD as a specific probability. The key thing in a dual agent setting is that you'll have a maximum tolerated dose contour now rather than just a single dose. But when you look at discrete doses, and often phase 1 trials, they don't want the flexibility of continuous dosing. Most of them still select discrete doses. Even if you have a contour, there might be one dose that is actually closest to your target. That's just the way it's set up. So we did a review of all the dual agent dose escalation type methods in 2013 with one of the authors is our PhD student, Graham Wheeler. So he spent his whole thesis on continuous, and this is a sort of model which Peter Thal had a 6-parameter surface model. So they're quite complicated pieces. You normally have 2 parameters on each margin, and then maybe 2 that describes the actual contour. But during his thesis, we realized that actually these models, there's quite a lot of problems. They give flexibility, but you're fitting now many parameters on a very small study, so you get problems with convergence. If you do anything like clever, like we set our PhD student doing Bayesian de-optimality in a full Bayesian way, and it just it takes months to do some simulations. And in the end, which model do you pick? I mean, people have lots of competing models, so can we do something better by removing the model? And often people come with, I've only got 2 doses on this drug, 3 on this, or 2 on this. There's just no point in having, say, 4 combinations and fitting a really 6-parameter model. So often things are much simpler situations. So we wanted to do a model-free approach to avoid that choice. We also wanted to make it conjugate. So if it's conjugate, then I can implement it in Excel. I can at least convince a clinician or someone to play about with a model in Excel. I'm not saying you can do everything in Excel, but it was just that sort of step to convince people to use it. So this is what our motivation is, to get this method that people understand, play about with, and hopefully pick up quickly. So the trial that we spoke to the statisticians in UCL Cancer Trials Unit was ORCA-2, and they were looking at head and neck cancer, squamous cell, and they were looking at, there's a standard treatment underlying this, so that there's radiotherapy and cisplatin, and they wanted to put a PARP inhibitor on top of that, which is olaparib. Now, the way our method works is, although we have discrete doses on both two drugs, you can swap dose for days. So they had 3, 4, 5 days of repeated dosing and level of dose on. So this also is a dual agent. We framed it as a dual agent. So I'm going to swap between days and doses and dose if we got drug or days dose. But it's the same problem. So they specified a TTL of 33%, and interestingly, because they got a lot of standard treatment, and a lot of people tend to do this, is they stick with the standard and add something in. Now, clearly, the standard is probably already at a maximum tolerated dose. So we do want to explore the surfaces more. I think there's a bit of resistance to do more experimentation, but I suspect we'll get side effects really quickly, and we might not be able to go very high up on the PARP inhibitor. So that's just a warning. Just we need to explore both doses, and actually, yeah, there might be some risks that you're reducing a standard treatment if you're adding in these additional treatments. So take the discrete setting of the two doses, dose A, drug A, and drug B, and say if we've got 4 levels of both drugs, then we have 16 probabilities, true probabilities of toxicity that we want to estimate. And the only assumption we're going to make is we're going to have strict monotonicity. If you increase the dose of one drug, you are going to increase the toxicity. That's our only assumption in this. And when you speak to clinicians, they sometimes think, oh, but maybe there's some synergy, and this might be lower than these. I mean, if we get into the realms where monotonicity doesn't hold, you'll be giving advice to escalate when you see too many toxicities. I mean, it just doesn't make any sense. So I don't think anyone's going to dispute this one assumption about monotonicity. Because it's a conjugate Bayesian analysis, I will have to specify a beta prior for every single dose combination. But and there's sort of simple ways of doing this. You can interpolate these, but actually it doesn't have that much influence. We believe when you start a trial that if you want all these combinations and you want to experiment on them, then they're probably going to be less than the target toxicity level. So as long as these are all less than the target toxicity level, you're going to experiment there. If you start putting these things, or this might be too toxic, then it's going to have some resistance to go there. So you have to have some good prior belief there. And we're having a very weak prior. I mean, if you add up all those A's and B's, they add up to 1. So that is pretty weak. So we want very little influence of the prior. So it's really a construct just to calculate posteriors. So in the ORCA trial, and when they came to us, they had this really complicated scheme of how they were going to do their trial. They were going to dose in different cohorts. So they're going to start here, add to 4 days, and then they were going to split into 2 cohorts, put 3 there, and then carry on splitting cohorts. So it was going to, so they kind of defined levels of equality of what they believed a priori, what the similar toxicity would be. So 100 mg 5 days is similar to 200 mg 3 days. So this is what they decided, and this is how they were going to do it before they spoke to us about PIPE. And actually, we took this diagram and just translated it into a possible prior. So these are all the sort of pairs. When I was saying 203 days is the same as 105 days, I've given them the same prior belief. So there isn't much preference between those. So that's the prior that we set up for them. And like I said, the target toxicity level is 33%. We're saying every dose combination is safe enough to experiment at the start. And how we do the standard thing about you have a beta prior for every dose combination. After M cohorts, we're going to have a number of DLTs, a number of people at every dose combination, and we're just going to do the standard posterior calculation, which is still a beta distribution where you're adding all your prior parameters with your number of DLTs and number of non-DLTs. So I now have a posterior distribution for every dose combination. They're all independent at the moment, and this is all standard stats theory. But the interesting part for us is actually when you think about if we have an estimated toxicities for every dose combination, we're only interested in the cuts between where you have a too toxic area and a safe area, which is defined by the TTL. So all these are above the TTL, all these are below. And it's actually, this is the maximum tolerated contour if that was the truth. And if we split this space into this contour, and that's really what we're interested in, then I'm interested in, I've just defined this as a binary matrix of dose combinations above, dose combinations below, and I'm going to use this notation later of above and below. But a first thing about the contour distribution. So just in a 2x2 case, there are 6 possible monotonic contours, which, yeah. And if you can work out how many monotonic contours there are. The space is a lot less than every single contour. But it's this contour, once we know which contours exist in whatever number of doses on each level, we now want to assign a probability to each of these contours and use the distribution of contours. Now, we just thought about, and in our paper there is a decision theoretic way of constructing the same distribution. But if you think about each distribution for every dose combination, all I want to know is this tail probability. What is the probability that the truth is less than the target? Now, the probability that the maximum tolerated contour is a specific CS contour, which is just the binary matrix, is this expression. So what we're doing is saying that if CS, i.e. your matrix is 1, you're above the TTL, I'm going to multiply by this tail, which is the probability your true probability is above the TTL. And then if your binary matrix is 0, i.e. you're below the contour, then I'm just going to multiply by the tail probability that your truth is below theta. So this is our distribution function, but obviously we want to restrict the contours to the monotonic contour space. So we're just going to normalize by adding up all those probabilities in the restricted set of monotonic contours. This is the hardest slide, but once you've got this distribution, I'll say how we're going to use it. So once we've got this contour distribution, we're going to pick when we did this work in the paper, we picked the modal estimate, i.e. what is the most likely contour, and then focus on where to pick doses conditional on that. We've also got, we also use the contour distribution for safety. So we use a sort of averaging over the contour distribution, which I'll describe in the next slide. So let's take a week prior on the 2x2 case. Our target toxicity level is 0.3. These are the 6 contours, and these are the 6 probabilities given these values. And you can see at the start, this is at the start of a study, so there's no data here. This is all the vague prior. And you'll see that the probabilities, there isn't much difference between the probabilities as you'd expect. There's no data there. There's a slight, this is the mode, this is the most likely, and that maps directly to those probabilities. This probability is obviously above the TTL, hence that's why this pattern is the most likely, but there isn't much preference yet. Now, how we do the averaging contour is if we think about averaging the probability of a toxicity over this distribution, if you think about the top left cell, it is toxic, toxic, toxic for those 3 contours, but not for the others. So we just add these up. So this is the averaging over the contour. So that's 0.45, which is the addition of those 3, which is probably what I got there. And you'll see even from the start that if I did the top right corner, all those 5 are above the contour. These are the probability of the contours. They all add up to 0.84. And we have a safety constraint that if any of these probabilities are above 0.8, we say it's unsafe. So, so this is the real crux, I mean, the distribution of the crux of using all the data in one. So we got lots of independent posteriors, but we're combining all the information into one statistic. So it is using the structure of your surface. So it's not, yeah.
This is all without any data?
This is purely prior. So it's got the weight of one person in a 2x2 grid. So it's, you know.
So already right from the start, one of the contours is...
One of the dose combinations is unsafe from the start. But then in my prior, I have said it is unsafe. So it is just purely reflecting in prior. So obviously, once we put in data, these probabilities change very quickly. Yeah. But I think it's important to have this sort of safety constraint. Otherwise, you need to be safe. Now, if we think about the contour that we select, which is the most probable, there are certain points on the discrete space that all these doses are the closest to the MTC. And these are the ones that we want to experiment on. Now, at the end of the study, if this was your contour, then these three dose combinations that are above the contour, there's no actual upper bound on their toxicity probability, so they could be unsafe. So we would never recommend those at the end of the study. We would only recommend the doses, the closest doses underneath the contour, because they are safe. And the other key thing, well, in this, you can see that because there's a step there, we're going to have two possible MTDs to recommend for phase 2 doses. And obviously, if this was just one step, you'd have 1. If it was loads of steps, you'd have like 5. So it is kind of dependent on the model. And with all phase 1 schemes, people tend to avoid dose skipping, so we've allowed that as well. So if we're currently at this dose, which is 3-2, we only allow escalation in a single dose level on each direction. So we're only allowed to pick doses within a constrained box. So this is our sort of dose skipping constraint. So how do we pick the dose for the next cohort as we're going through this? So we calculate the set of closest doses, or dose and day combinations, and we pick the closest with the smallest sample size. Now, it's a very simple rule, this. It works very well. And part of the intuition is we want to experiment across the contour. And if obviously you've got 2 dose combinations that you've got, say, 2 patients on each, then you would just pick them randomly which one to pick. Now, sample size here is not just the number of people you have on each dose combination, it is the combination of the prior as well. So that gives a slight preference to dose combinations. But I mean, if you have a higher preference, you just make your prior belief stronger. And when you think about where the closest doses are and that sort of dose skipping constraints, you just see whether they overlap, if there's any closest doses in the box. If there isn't, if you're allowed to diagonally dose escalate, you would pick that one. Some people think going up 1 level on both doses might be too controversial, so some people remove that as well. But we've implemented both. Now, the key thing about when you don't have a model, if you don't experiment on either side of the contour, you don't learn about the contour. So you really do need to experiment on both sides to move that contour. Obviously, if you had a model, this is where a model can be beneficial. If you knew a model and it was correct, you would be extrapolating into that area that you haven't experimented on. So we just have to specify that. So I showed how we pick the prior for ORCA2. I don't think it's that difficult a choice. I mean, people could draw MTCs, the contours, or pick the highest and lowest points and extrapolate. There's loads of different ways you can do this. But if you ever pick a dose combination above the target toxicity level, because we don't have a model, it is going to take longer to overwhelm that because of the safety constraints. So we wrote some R code, and this is some of the output from an R code of a single simulated trial. Here we're looking at cohort sizes of 2. We start in this cohort of 2 patients. There's no DLTs. We do a diagonal escalate. So in the second cohort we go up, and at the moment the black line is our prior belief that every dose combination is safe. So our toxicity contour is just on the edges. You carry on until this fourth cohort where we saw 2 DLTs out of 2. So that's like 100% toxicity rate. And immediately the contour removes that dose combination as being too toxic. And then we carry on. And in this simulation, we're always on, we got a local neighborhood constraint. But so we go to this point, and there's 1 DLT out of 2. So again, that's going to be above the contour because it's 50%. So the contour moves again, and we carry on experimenting, and you can see how it sort of spreads out. And the last contour has 2 doses that are recommended as the MTDs. I think the green was the truth, or I can't remember which one. But that's a simulation of 1 trial. So to prove that our method worked, we took a paper by Braun and Gier in 2013, and they had a whole bunch of simulations comparing the generalized CRM, copular regression, and their latent contingency table approach. And we set up our simulations to be exactly like theirs. So they had a maximum sample size of 50. They had a cohort size of 1, which is really bad for our method, but we still went with them. And no one, I'm not sure if you get 1 toxicity out of 1 person, you're really going to be in choppy water. So I don't think that's a good thing to do. We did 2,000 simulations, and it essentially takes 2 minutes to do that in pipe because everything's conjugate, so it's all very quick. Our target toxicity level was 20%. So they had lots more scenarios, but I'll just highlight 4. This was their prior scenario, so there's 4 levels per each group. There's our prior. They had a safe dose combination, so hardly any toxicity, so all of these are lower than the target. They had a toxic scenario where everyone's quite high quickly. And they had an interesting scenario where drug B didn't really increase toxicity by much, drug A did. So this is kind of like a sort of flat plane. And the good thing is about our method, you could pick any prior. It's a very difficult, and we found this with one of our previous papers, of comparing different dual agent models. If you're doing an Bayesian approach and a model-based approach, then your priors, you can't match your priors between different methods. It's incredibly difficult. But we can because it's non-model based, we can compare to any method. So we can set up this prior, those are just the probabilities. So if we came to the first, also we took their reporting statistics, so what they recommend and what their experimentation. And most of these methods only look for 1 maximum tolerated dose. So they were interested in targeting directly at the 20%. And if you look back, there are, in each of these scenarios, that would be the truth, that would be the truth, no truth, and that one. So you can see, if that's the MTD, with us, with a contour, we're going to definitely select more dose combinations that are similar. But we stuck to their statistics, and you'll see that our method actually performed very well if the prior equals the data. So we recommend 98% of the time within 10%. The experimentation was slightly better as well. So we kind of outperformed the models when prior and data agree. In terms of the safe, we didn't perform so well. This is partly because for us, we have to experiment one step at a time beyond the contour. So we're constantly doing a lot more experimentation. Whereas in a model, in a completely safe dose combination, you can extrapolate right to the corner very quickly. So there's no way we can outperform them, although we do have some recent work where we can actually improve this. This is also partly having cohort size 1 makes us much slower as well. In the toxic, they targeted 20% slightly more than us, so it was slightly less safe because we equally don't have a model to predict certain moves beyond the contour. But its recommendations were within similar if you add up those two numbers, so 90% within 10%. But we did experiment a little bit over as we have to really. Obviously, if you have a model, you then say in these dose combination areas, this is unsafe, I won't go there. That's what a model does. And in the last scenario where drug B was safe, we actually outperformed all the other methods by quite a long way. And that's because the data, because it's quite a flat plane, steep in one direction, shallow in the other. Most of the other models are sort of curved models. So as soon as the model doesn't actually fit the data, their performance goes down quite a lot, whereas for us, it doesn't make any difference. So we gain something once the model is incorrect. We've also started looking at time to event extensions to this. There's a few different ways of thinking about if you want to recruit another cohort while you're still observing the previous cohort. And most of them use some sort of method of partial information. So Ken did a paper where if you've got 70% follow-up of a single person, you would say, and you haven't seen a DLT, you would say there's no DLTs from 0.7 of a person, and you can put that in his data. We can easily put that into any of the posteriors that we're constructing as well. Anastasia Ivanova, I don't know whether this is actually published yet, but she thought of a more conservative way, where she's saying the last 30% of that person, they're sitting in a DLT, so they give 0.3 DLTs of a whole person. So I think there's lots of different assumptions that you can do on how you give partial information, different weights. But I think whatever assumptions you make, it's going to be quite an easy way of introducing it into the pipe method. And so Graham Wheeler, our PhD student, is looking at this problem, and he hopes to present it in Turin in September. So, and just like I said, there's some R code, and we're not allowed to use the name pipe, because obviously pipe is a generic computer name, so it's called pipe.design. I've got some Stata code, and there is an Excel spreadsheet available as well. So I wonder... So part of our collaboration with Roche, so when we presented it to Roche, Chris Harbron, who's a statistician there, picked up the method and wanted to introduce it into their trial. And actually, he actually took our R code and made a Shiny app. I don't know if anyone knows about Shiny apps. It's just a way of doing an interface via a browser. So he's written the interface. This isn't on CRAN R yet, so we're still thinking about various things. But it is, you can pick how many dose levels on each drug, all the toxicity target level, cohort size, and different strategies and the safety constraints. So there's various things you can select, and then he has another screen where you can enter data. Well, you can set up your prior by either selecting what value there is there, and then pressing the interpolate, or just having completely flat priors, and then you can add in data as you go along at certain levels and say that there's no DLTs. And so the black lines in all these diagrams is actually the beta distribution, the posterior beta distribution. And he's got a method where we actually sort of use the contour distribution to give a smoothed more like a PDF of this distribution. So this is a little plug. He's talking about this at PSI in Berlin. So he will talk in more detail about these diagrams. And yeah, we use the modal estimate, but he's developed a median estimate and various different ways of describing the data. So that's 5 minutes, great. This is my Excel spreadsheet. So in this one, you can, I have to remember, don't I? Yeah, so this is our priors are set there. Oh yeah, so there's a button. So our priors are actually, we pick the A and B so that the median of the beta priors equal whatever you specify. And you can just put in data here, and then it will tell you what's the most likely contour given the data. So this is just that prior that I had on a few slides ago, or roughly. And it'll tell you the probability of each of the contours. So if this is the most likely, this is where you start making your choices. What I can't program in Excel is how to suggest which doses to give. So I kind of sort of did an Excel spreadsheet so you could kind of intuitively understand what's happening, which I'm happy to give to anyone. So that's the interface. So just in conclusion, so we believe this method is really simple and accessible because every time we speak to people, people do tend to understand this, and it performs not bad even against some of the models. Clearly, if a model is correct, then the model is always going to perform the best. So our thing is, and a lot of clinicians are very skeptical about models, so we're trying to remove the model to make it a bit more, so people can implement it better. Like I said, it performs very well when the prior is actually the truth, and there is a cost about non-model based. You need more experimentation because the models do some of the filling in the gap. So yeah, I think that's all I've got to say. Thanks.